And now we'll move into section 2. So I'd like to introduce Dr. Zeynep Kosaloglu-Yalcin, our cancer bioinformatics instructor. She'll be giving back-to-back presentations, another mammoth run, but the first one will be the Cancer Epitope Database and Analysis Resource, also known as CEDAR. Thank you, Zeynep. And so maybe while we do that, I can give a little bit of background. CEDAR is is what we call our sibling resource to the IEDB. And you'll see there are some visual similarities and some key differences, which are really important. And where the IEDB is funded by the National Institute of Allergy and Infectious Diseases over the last 20 years, CEDAR is actually funded by the National Cancer Institute. So a different funding source, and it's only come about in the last few years. So now I'll hand it back over to Zeynep to talk us through what CEDAR is. Thanks, Zeynep.
Thank you, Nina. Yeah, thanks for the introduction. So I gave this presentation the last 2 years now also. So we're always updating our presentations, and I'm showing you the latest updates to our database and the analysis resource. So this year, I'm going to talk mainly about the analysis resource. So we have a lot of new exciting developments there. So I'll start with a brief introduction about what cancer epitopes are. So cancer epitopes are short peptide sequences derived from tumor-specific or tumor-associated antigens. And these cancer epitopes are very important. They are crucial for the immune system's recognition and the targeting of these cancer cells. So the identification and the study of these cancer epitopes is important for us to understand cancer immunology and also to develop immunotherapies. So there are several different types of cancer antigens, such as the overexpressed antigens or the tissue-specific or the tumor-specific ones. And also we have these so-called neoantigens that are the results of mutations in the tumor DNA. So as Nina mentioned, while the IEDB hosts a wealth of data for different diseases like allergy, infectious disease, and autoimmunity, cancer is not part of the IEDB. It never was. So we received funding from the National Cancer Institute to develop a sibling database specifically designed for cancer. And just like the IEDB, CEDAR is also divided into two main components. We have the database, where we catalog all the relevant biological and clinical data for these epitopes. And then we also have the analysis resource, where we provide computational prediction tools and analysis tools. So the curation of these cancer epitopes follows the same processes that we use for the IEDB. The only difference is that we added specific keywords to our query that is performed biweekly, semi-automatically. So we added specific keywords that specifically cancer-related epitope data can be retrieved from PubMed. And when we look at all the papers that we retrieved, around 13% of the papers were found to contain cancer-related epitope data. So we started curation for CEDAR around 3 years ago, and we started with papers containing neoepitope data, because as I mentioned, these neoepitopes are highly relevant for cancer immunology and research right now. And neoepitopes is a very broad category, so to say, because it covers many different antigens and a lot of different cancer types. And in contrast to that, we also curated prostate cancer epitopes. So this category is very limited to one cancer type and a few antigens only. So curating these two categories first really helped us to fine-tune and optimize our curation processes and also the database itself. So after that, we started curating papers based on the date of publication, and we started going back in time. So far we went back until 2015, and we are up to date on most of the for most of the years. And so far we have curated almost 2,000 papers. So our curation team has been very busy, and I think it's very impressive for the amount of time. So this is the website for CEDAR, for the CEDAR database. It looks very similar to the IEDB website. We have a CEDAR-specific color scheme of purple, and then we also have some distinct features here in CEDAR. So for example, here we can select the type of antigen we want to search for. So neoantigen, viral antigen, also germline or self-antigens. And we also have the cancer panel here, where we can select a specific cancer type and also the stage of the cancer. And we can also select if the disease occurred naturally in the patient, or we have an animal model, for example, or if the immune response was observed as response to a vaccination. So just like in the IEDB, you also have the three main assay types in CEDAR, namely the T-cell assays, the B-cell assays, and the ligand elution assays. On this slide, I just wanted to briefly summarize the amount of data we have currently in CEDAR. For example, here for the T-cell assays, we have over 44,000 unique peptides from 12,000 antigens. And then of these 44,000 peptides, around 13,000 were tested positive. So they were tested and recognized by T cells as epitopes, actually. Yeah. I just want to briefly show an example query to demonstrate CEDAR's functionality. So let's say we want to search for a new antigen data that were tested with T-cell assays that had positive outcomes and in lung cancer. So what we would do is we select the new antigen here and deselect the other options. We select the human as the host. We deselect B-cell and MHC ligand so that we only get the T-cell assay results. We select only the positive outcomes. And for this example, we want to focus on the class 1 restricted epitopes. And then here we have a disease finder. So if we start typing lung, we get all these suggestions. So we would select lung cancer here to search for epitopes specifically from lung cancer. Once we execute this query, we get this results page. Here on the top, we have the filters that we use for our query. And then we have the different tabs here, listing the epitopes, the antigens, the specific assays, and the receptors that were reported, and also the references that reported the data. So here we're looking at the epitopes tab. We have the epitope sequences, the antigen it was derived from. And because we search for neoantigens, the antigens will always have the prefix of neoantigen, indicating that this is a mutated antigen. And then we have the host, we've filtered for human. And then for each epitope, we have the number of references and assays available. So we can get more details about an epitope by clicking onto its CEDAR ID here, which will take us to the epitope summary page. And here we have now access to the variant information. As I mentioned, neoantigens are the result of DNA mutations in the cancer genome. And exactly that information is listed here. We have, for example, here the specific base change that happened. The T was mutated to a G, and the location in the genome here for, in this example, chromosome 7 at this position. We also collaborated with the Open Cravat team in Rachel Karshin's lab to even get more information into CEDAR. So Open Cravat links us to all the other external databases that contain any relevant data. So here, for example, we are displaying the dbSNP ID, the damage prediction of the mutation, and then we also here have the clinical significance. For example, this specific mutation is known to have a drug response, and then here also we have the different cancer types that were observed in other studies. To even give it more information, we have this link out to the Open Cravat page. So they created this variant summary page for us, where we have now access to even more external databases, cancer-specific databases, prediction tools that were run on the mutations. So this is all information about the specific DNA mutation. Yeah. Okay, so now I want to talk about the cancer-specific tools that we developed for CEDAR so far. So CEDAR tools are also available as part of the next-gen IEDB tools. For example, when we click on the tools here, on the drop-down menu, we can see the list of tools, and then everything that is related to cancer will have this little CEDAR logo next to it. And we can also go to the all tools list by clicking on this link here, and then there we can filter for the cancer tag to get all the tools that are cancer specific. So the first tool we developed answers the question, what new epitopes are generated by a given DNA mutation? As I mentioned, the cancer genomes have specific DNA mutations at specific positions, and when these mutations are translated, they become translated into a mutated protein, and when this protein is then being chopped up into all these mutated peptides. So researchers often have their list of DNA mutations in the form of a VCF file that they got from their genome or exome sequencing. But what they are actually often interested in is the list of the mutated peptides. So we developed a tool to exactly answer this. And we named the tool Mutated Peptide Generator. The user simply uploads the VCF file containing the DNA mutations, and the tool then outputs the list of mutated peptides. And the user can also specify the desired peptide length, the mutation, the position of the mutation within the peptide, and the overlap of the peptides if we have longer frameshift peptides. So this is how the results look like. We have our input information from the DNA, the chromosome, the mutation position, and the base mutation. And then here we have our annotations, and we get the peptides here that are generated. So we have the mutated peptide with the mutation. So we selected the position 5, and then we also have the corresponding reference file type peptide. So when we are dealing with new epitopes, we also always have these two peptide pairs. We have the wild type and the mutated peptide, and researchers oftentimes want to analyze these peptides side by side and compare them. So they want to know the binding affinities, how do they compare, looking at the mutant and the wild type, and what specifically is the difference between those two peptides. So for that, we also developed a tool named it the Peptide Variant Comparison Tool. Here, the user can input peptide pairs and then select the MHC alleles of interest, and then also the prediction tool. So right now, we have all the MHC class 2 prediction tools available to select. MHC class 2 will be available in a future release. And right now, we have all the different tools here. In this example, we selected an MHC pen 4.1. And we have our list of peptides from before that we generated. And so we get a very nice results table where we have our peptide pairs. And then we have the prediction scores side by side. And then we also have a column displaying the difference between the two. So this is really helpful for researchers to have everything in one place and directly have access to the comparisons. So we also provide scatter plots here. When you click on this tab up here, So this scatter plot, looking at this, we can easily compare the prediction results for our peptide pairs, and then we can spot any outliers that might be of interest. For example, if the mutated peptide, we have some examples here, they bind much better than the wild-type peptide, for example, those might be really interesting to use for immunotherapy. So in a study we published back in 2002, we showed that integrating both MHC binding and the antigen abundance improves the prediction of epitopes. And we showed this for both for cancer neoepitopes and also for viral epitopes from SARS-CoV-2. So we took these findings and we also implemented the findings into a new tool named XLF. And XLF actually stands for Antigen Expression-Based Epitope Likelihood Function. And right now, this is still available on the IEDB legacy side. So this is very similar to a regular MHC class 1 binding prediction. So we have we input the peptide sequence, the MHC molecule we are interested in, but now we also have to input the a gene TPM, which is the expression value for the specific source protein of the peptide. And the result is a combined prediction score. So the user can upload their specific TPM values directly in the table, like here. But also we provide pre-calculated expression values from the Cancer Genome Atlas, where they can select specific cancer-specific TPM values. So the result again looks really similar to the MHC1 binding predictions. We have the peptide and the allele, but now we also have the TPM that was used, and then we have our XLF score, which was shown to outperform the regular MHC1 binding predictions. So when we were developing XLF, we noticed that expression values are oftentimes not available for patients. And we used TCGA as a public database to retrieve expression values from. So in another study, we wanted to formally investigate how these public databases can be used. So we showed that the gene expression from these public databases correlates very well with the individual RNA sequencing data. Of course, there are specific differences for each patient, but overall, there is a really good correlation between the expression data. So we derive from that the public databases can be used if the patient-specific data is not available. So we also found that some peptides, they can be derived from different source proteins, and it is not clear which expression value do we use in such cases. So here we have a nice example, and we have, for example, here a DNA sequence with the different exons. And as you know, if this depends on how the exons are spliced and together, we can get different transcripts, and then these different transcripts can get translated into different proteins. And then if we have a certain peptide, for example, here we have this red peptide that occurs in 2 of these proteins, and then it also occurs in a different gene in 1 of its proteins. And then we have peptides that only occur in 1. And yeah, we also saw specifically when we look at gene families that share a lot of sequence homology, that a specific peptide can occur in up to like 10, 20 different proteins. So it is not clear what expression value do we use in such cases. And we formally investigated it, and we found that summing up the different expression levels from the different source proteins provides the most accurate results. So again, we took all these results and put it into a tool and named it PepX, standing for Peptide Expression Annotation. So with PepX, users can look up their peptide expression levels from several public databases. So the user inputs simply the list of the sequences they're interested in, and then here we have our list of public databases that we pre-processed and made available. So we have the Cancer Genome Atlas, TCGA, or the Cell Line Encyclopedia, CCLE, but we also have healthy data such as the Human Proteome Atlas or GTEx, which provides tissue-specific healthy expression data. So this is then how the results look like. We have the peptide, and then here we have the list of gene symbols the peptide was found in. And this is a nice example. It was found in a lot of different peptides, but here, as we can see also from the name, this is a gene family, presumably sharing a lot of sequence homology. So the total peptide TPM would take the TPM, the expression values of all these proteins, and then sum them up and provide this data here. So the CEDAR tools are also available for our new pipelining feature. So in the top left-hand side of the IEDB NextGen side, we can build our pipeline. So here I have an example. For example, you would first take your VCF file and generate your mutated peptides. And then you can use the peptide variant comparison tool to run side-by-side predictions. And then you could also use PepX to retrieve expression data for all of the peptides. So there are a lot of ongoing activities at CEDAR right now. So one project that we are working on is the manual benchmarking of prediction tools for cancer epitopes, because there are a lot of tools available to predict epitopes. Many of them are designed for viral epitopes, so we are also trying to see how well they can predict cancer epitopes. So I showed that we have the variant information available on the epitope summary page, but we are also working on making this information available for download also, and also to make it searchable and more accessible to the users directly from the website. Kelly just showed us the API to access the IEDB data, and we're also working on an API to access the CEDAR data. And we also want to include more of the CEDAR tools in the IEDB NextGen Tools website. And exciting news is that we also are planning a CEDAR-specific user workshop for Q2 in next year, which will be a shorter webinar, but then we will cover everything related to cancer and CEDAR tools and the website as well. So we will also be attending different conferences next year. So we will be at the annual Association of Immunologists conference in Hawaii, and then we will also be at the American Association for Cancer Research conference, AACR, in Chicago, and these are our booth numbers. We always have some curators there that can answer all the specific questions, and they can also run demos for you if you have any more questions. So to summarize, CEDAR is the cancer-related sibling resource of IEDB. So our curation is ongoing and it continues rapidly. The data is growing. We have now variant information available for neoepitopes, and we are also working on making it easier accessible for users. And we have now finally several cancer-specific CEDAR tools available on the Next Generation IEDB Tools website. Okay, so that's it for this presentation.
Awesome.
And I can go over to the next one.
Thank you so much, Zeynep. I'll give you just a minute to breathe and switch over and to say thank you for that. So About 25% of the people that registered for the workshop this year had attended in previous years. So if you are one of those people today, you may have heard Zeynep's talk last year or the year before on CEDAR, and hopefully you've seen some developments over the last few years. And in particular, we talked about the analysis resource coming soon. And this year we're super excited to have shown some of those tools, PepX, mutated peptide generator, those kinds of things. So yeah, that's exciting to see the development over the last few years. And we'll be sharing the links on Twitter as well for how you can access those cancer-specific tools. But we are utilizing the Next Generation Tools platform, which you would have heard Jay and Dan talk about yesterday if you had attended. So we're really creating synergies between the IEDB and CEDAR resources.